Good evening, everybody in the Americas. A very late good evening to those in Europe, and a early morning to those in Asia. We've come to our third check-in for our global meeting. As promised, I brought a special guest to this session. If Danny can have his kids be special guests, I thought I'd bring Catherine here. Catherine, can you wave to everyone around the world? So Catherine and I are happy to be here to provide you a tremendous update of what's going on in this community. Do you want to actually put your toys down, Squeaky? And you can sit with me? Well, I think you all will be way more interested in the update of the Odyssey community's progress than Catherine has been. There we go. So believe it or not, it has only been 32 hours since we started this study-a-thon. If you remember, at 3 a.m. Eastern time yesterday, Danny kicked us off and we walked through a proposal of what we were going to do. So the question is, what progress have we made just 32 hours later? Well, unfortunately, we have to put this in the context of what's going on all around us, and it's been a pretty grim day, particularly for those of us here in the U.S. There is over 590,000 confirmed cases around the world. The United States just broke a regrettable marker of having over 100,000 confirmed cases in the United States, making it the most prevalent country around the world. We're now dealing with the aftermath of having over 26,996 patients now succumb to this disease. So this is continuing to accelerate as an important problem, reaffirming the focus that we have. And it's really important for us to continue to keep perspective about why what we're doing is so important. So it's really uplifting for me to see how much progress we've been able to make in these 32 hours. So for those of you who may have missed all the activity, I want to highlight this website that Craig Saxon has created, odyssey.org/COVID-19 updates. You can see all of the material of what's happened in the last 32 hours, including reliving the videos that have provided the last updates. But just to summarize in bullets, what have we actually done? Well, in only 32 hours, we have convened 351 collaborators from 30 countries into this community forum that we're working together. This is the 6th of our global huddles, where we've had somewhere between 50 and 100 researchers coming together to talk on 3 different continents every 8 hours. We've now engaged 15 concurrent channels where we've been having hundreds of conversations with each other throughout all of these topics. We've collaboratively reviewed over 2,000 publications through our literature review work to try to summarize what's known about this important diseases. There are now 6 draft protocols that are available to the MS teams to review. But it's the last 2 that I'm going to highlight and really spend some time to focus on. As a community, in 32 hours, we have designed 208 cohort definitions that are now being thoroughly evaluated and prepared to serve as the input of all of our studies. And I think probably most incredibly is we have now assembled a distributed data network that has 36 data partners who have signed on and prepared to use their OMOP Common Data Model instance and execute the analysis packages generated by this community to allow for us to generate evidence that can support this global cause. So what I'd like to do in a few minutes is just summarize the ups and downs we've made in cohort definitions and really dive into the tremendous work of the data network that we've put together. So let me first start by going to, here we go, here. I want to go right here to show you the odyssey.org/COVID-19 updates page. If you haven't gone there, I want to give a shout out to Craig, who's provided a quick rundown of the highlights. For those of you who missed it earlier today, we had a credible announcement from Korea providing access to the national claims data from HERA as a mechanism for us to generate evidence and to learn from the experience in Korea and bring that around the world. So congratulations to Chan and Ray, and thank you to Hira and the Korean Ministry of Health for their collaboration. What I'd like to highlight is first the ups and the downs. The ups that I'd like to show you is to remind you that we have a development environment at atlas-covid19.odyssey.org that is serving as our collaboration place for us to design and evaluate cohorts. And as you can see on the screen here, we have now, as a community, collaborated to generate 208 cohort definitions that have been brought together to try to provide an approach to create populations that we can study. And this has spanned all of the work necessary for us to design our characterization, estimation, and prediction studies. And there's quite a lot of work that's still to be done, but it's amazing to see us come together in 32 hours to produce 208 definitions that we want to explore thoroughly. Now, that's the good news. Hold on a second, Squiggy. I got to tell the world what we're doing. So the good news is that we've created and drafted 208 definitions. The bad news is if I go to atlas.odyssey.org, that is our production environment where we're putting all of our final analyses. You'll see that we have no final estimation specifications. We have no final prediction specifications. We have no final characterization specifications. And for those of you who are observant, you're going to notice that the number of final cohort definitions we have is actually smaller today than it was just a few hours ago. So what's happened? Well, actually, it's been a tremendous learning on the front of creating phenotypes that can run across our data network. Our phenotype development group has been hard at work evaluating phenotypes, and what we're learning is that there's a lot more nuance to designing and evaluating symptoms associated with viral diseases, and to come up with appropriate definitions that allow us to explore influenza, flu, the flu-like symptoms, including fever, cough, dyspnea, and pneumonia. I myself have spent a great deal of time with Danny and Christian and Anna and the entire phenotype team wrestling with the questions of how do we actually find the right trade-off between sensitivity and specificity of these definitions. But what I'm really excited about is the progress that our phenotype team has come up with. We now have cohorts that are just pending the review to move from development environment here in atlas-covid19 into our production environment. And our real target is to try to see as many of those cohorts move over tonight as possible. What our expectation for this work will be is that once we have cohorts that are finalized, we'll be able to also finalize the study packages that have been drafted by all of the work channels. And it's actually exciting to see that most of the analysis decisions have already been made for all of our prediction and estimation studies. And on the characterization front, there's been a great deal of work that's gone into, now that we've defined our cohorts, thinking about exactly what characterization features we want to entail, and we've made tremendous progress on that. So while we've actually seen that we have more work to go in terms of coming up with definitions that we feel confident of in running across the network, I'm really excited by the progress we made, and I feel much, much more confident in the quality of the cohort definitions that we're creating now, based on the fact that we have lots of collaborators all looking at the definitions and using our entire data network to really understand exactly how do we phenotype these diseases appropriately. So I want to give a great shout out to the phenotype group for all the tremendous work that they're doing. But the real thing that I'm most proud of and excited to highlight today is our data network. There's been an awful lot of questions that have come up throughout the last couple days about what exactly is the Odyssey data network. We talk about the fact that anybody can be part of the Odyssey data network. All it takes is a willingness to use open community data standards, including the OMOP Common Data Model and the standardized vocabularies, and the desire to, where appropriate, use that data to answer questions and generate evidence across the network. As you all know, we're a distributed data network, so we're not looking to share patient-level data across institutions, but rather send analysis packages to the data and allow sites on a voluntary basis to execute that code generate aggregate summary statistics, and send those results back to a central study lead so that those results can be compiled across the network and summarized for our final results. And what we've seen time and time again is that the more databases you have access to, the greater confidence you can get in your results, because you're no longer looking at the peculiarities of one population, but rather you can get the breadth of knowledge that can come across an entire network. So we talk about the OHDSI data network in broad strokes as anybody who could do any studies in any capacity. But what's the OHDSI data network look like for this Study-A-Thon and for our collaborative efforts towards COVID-19? Well, Kristen Koska has done an amazing job leading our study execution channel. And one of the things that she worked together with George and Jose and Talita on was making sure that we had an up-to-date tally of the data partners who are part of our Study-A-Thon, who have access to data, who have standardized their data to the OMOP Common Data Model, and who are raising their hand to answer the call of executing study packages and generating results. And I think the results of this compilation are pretty stunning and humbling and exciting to me. What you can see on this spreadsheet, which is available on the study execution channels, is a list of the data partners who are participating in this activity for us. In total, we have 36 different databases with named individuals who are ready to execute study packages on their data and share results. Those 36 databases represent data from South Korea, the United States, the Netherlands, France, Germany, the UK, Japan, Spain. We have data across that's 8 countries on 3 continents, representing both administrative claims and electronic health record data and hospital billing records. We've got data from general practice and data from linked between outpatient and inpatient services. The vast majority of these data sources have all adopted the Open Data Community Standard, which is the OMOP Common Data Model version 5.3. And on the basis of that, we know that we can design code as a community, share it with all 36 of these data partners, and have a high level of confidence that our analysis code will run and generate results and be able to share results back. Now, a question that I've seen several people pinging each other on the channels about is, okay, we've got data, but do we have any COVID data? Which databases have recent information? And I think it's really exciting to be able to enumerate the idea that we actually have 7 databases who are volunteering to participate in part of this effort that actually have recent enough data to contain patients with COVID that we can learn from. We mentioned earlier that the exciting announcement from Korea that the HERA data is now available to be run as part of this distributed data network, and for us to submit the OHDSI packages and generate results for our characterization, prediction, and estimation studies. But what should also be highlighted, and Ray Park posted some of the numbers, is that there are 2 electronic health record data sources from Korea that also have COVID positive cases. And we can actually use that data. And I think it's tremendously valuable for us to think about how the administrative claims data from HERA in Korea can be complemented with the perspective that an electronic health record system can provide. And so even though we have 2 databases or 2 sources coming from the same country, the fact that it's a different data type provides a tremendous strength that is kind of unparalleled around the world. I'll also highlight that in the United States, tremendous work has been done by some of the leading academic medical centers to really do some miraculous work to try to make their data as up to date from their electronic health record systems as possible. So Andrew Williams has volunteered to use the Claret data from Tufts as a mechanism to share what is the latest learnings that are coming from Boston. Thomas Falconer, Matt Spotnick, Karthik Natarajan, and George Ripsack at Columbia have prepared the Columbia University Irving Medical Center data to be able to be used, and have done a remarkable job integrating the new Epic installation at New York Presbyterian Hospital and trying to take that data so that we could access the most recent information about COVID patients that are experienced in New York City. We have data from Stanford University and able to look at the electronic health record data that's being captured in that tertiary care setting in California. And then also Adam Black has been graciously volunteered to collaborate with us using data from the Maine Medical Center. Now, what's exciting about this is we have 36 different databases in 3 continents and multiple countries. We have a breadth of legacy of data representing, going well past over a decade, but we also have some of these databases that are providing us a current lens into what's going on in our current pandemic. And while I'm convinced that this data will probably not be sufficient for us to answer all the questions we want, I'm excited that this provides us a breadth of data on 2 different continents that's going to allow us to get some initial insights about what's possible and to allow us to get some initial reads of some of these critical clinical questions that we're facing, and also is going to provide us the opportunity to know that we can develop analysis packages that will run, and as data accumulates and we grow the sample, we will be able to produce more reliable and precise estimates of all of the questions that are mattering for public health right now. So I want to thank and acknowledge all of the data partners who are part of our network for volunteering to participate in this effort. We recognize entirely the considerable effort it takes for you to take on the energy of adopting an open data community standard. I know it's not easy when you've got lots of people out there having lots of demands of your data, and to do something that's part of this voluntary network is really going above and beyond the call of duty. But I think it's this study-a-thon in this current situation we are in with COVID-19 that demonstrates exactly why it's so important that we're doing this. The idea that none of us could have possibly conducted anywhere near, if we had only our data by ourselves, we wouldn't be able to answer the questions we need. But together with 36 databases in multiple countries across 3 different continents, with 7 databases having active access to recent cases of COVID, we have a real chance to make a real big difference for public health. So I want to thank all of the data partners for making this generous contribution to be part of our distributed network. I want to thank Kristen Koska and Talita for leading our study execution as we develop our study packages over the next probably 8 hours. We're going to be leaning heavily on these data partners to be able to take those study packages and execute it against the data. And we've got Saturday and Sunday to see how far we can get with executing these studies, compiling the results, and then deciding what have we learned that we can actually share with the public to try to advance the knowledge that we have about this disease and the potential treatments that can ward off this illness for patients. So thank you, Kristen, thank you, Talita, and thank you to all of the data partners that are participating in this network. So what's the journey ahead? Well, we have done all of those things in only 32 hours. We've created cohorts, we've designed analyses, we've started protocols, we've reviewed the literature, but now we're leaning into a step where what we need to do is, now that we've started to design these phenotypes and evaluate them, we need each of the study teams to actually review their phenotypes for their specific questions, make sure that what the phenotype group has done is consistent with what the protocols have specified and that is the desired intent of the analysis. The good news is that the protocol development work has been going on in parallel with the study package work. And so what we're hopeful for, and it looks like it's working out quite nicely, is many of the groups have study packages that are tentatively prepared, and they're just looking for an input to be the cohorts and the phenotypes that our phenotype group has been working on. So in the next 8 hours, I think the marching orders for all of our channels is pretty much the same. Whether you're working on characterization, prediction, or estimation, I want you to be standing by to review the phenotype definitions as we make them available. And I want to see those phenotype definitions work their way into the study packages, and I want to see finalized protocols. Several of us have had several conversations, both internally within the community as well as external, about the visibility that this work deserves and it is already getting. So I think it's very important for us to recognize the responsibility that we have to write high quality protocols that specify our analysis and to try to follow the scientific best practices that our community has developed. You know, sometimes when we go through these processes of doing study-a-thons and we're working fast and we're working quick, some people have speculated that it's a bit of a gimmick that you work fast and then somehow what you're doing is you're compromising quality in the interest of speed. I just want to directly address that head on and say that we absolutely should not be sacrificing quality. We need to be doing high quality science, but it is not necessarily the fact that going fast means going poorly. What I've seen across the community actually isn't about going fast. It's about having tremendous talent and complete focus on a task that's allowing us to develop the work that we're doing with such efficiency. And at every step of the way, if there are issues that we need to address, we need to take a step back and evaluate it, just as we've done today with our phenotype definitions. At the same time, I don't want us to find ourselves in a situation where we're going to let the perfect be the enemy of the good. As I shared earlier, we now have over 500,000 patients inflicted with this disease. In the United States alone, we've crossed a threshold of over 100,000 patients. Patients are literally waiting for knowledge about this disease. Healthcare professionals are waiting for knowledge about effective treatments that can be provided. Policymakers are waiting for evidence to guide and inform what needs to happen for the public health. And so while we absolutely need to hold ourselves up to a high quality standard about how to do high quality research, I also want us to think critically as a community about how we deliver high quality science with high efficiency to meet the needs, not just of our own research interests, but meet the needs of the entire public who are waiting for the evidence that we're trying to develop. So we've made tremendous progress in the last 32 hours, but what I want to encourage you all to think about is not where we've been, but the journey ahead and how we can all focus on delivering high quality science as fast as we can to generate the evidence we need to support this pandemic the best way we know how. So with that, I will stop and take any questions that you may have. I see a couple questions have already been posted in the Q&A, so I'll take a look at those. And so I'll get started here. Question number 1, super important. Can we vote for who has cuter kids? Since Danny's not on tonight, I'd have to say that Catherine is a landslide winner. Though I'd have to say that what I'm impressed with Danny is he's got several of them running around each way, and he has not actually used them as a prop. They volunteered to be a prop. So there you have that. Well, I'll throw the gauntlet down to Danny to see how he can make his kids even more adorable in 8 hours. Another question was actually about this data sheet, and I want to pull this up here. The question is, does the red highlighting have something, what is the meaning of the red highlighting in the sheet? And it's actually a good question. So several databases that we have within our network are actually data sources that can be licensed by multiple institutions. For example, Christoph has access to the IBM market scan data, and Janssen also has access to the IBM market scan data. So the databases that are colored in red simply represent situations where multiple institutions have access to the same data asset. We've talked about this several times outside of the Study-A-Thon, but I'll just reinforce the point. I actually see this as a tremendous strength, not a weakness, because we're actually in the situation where even though it may be the same or similar data, the fact that we can have multiple researchers be available and be able to volunteer to contribute their data is a tremendous testament. And if we can run analyses across different databases, we clearly are not going to double count an analysis from multiple databases, but what we can do is divide the work across our network and also actually just make sure that things like our ETL processes were consistent. So if I run an analysis on my data at Janssen and Christoph runs his analysis on MarketScan at New Mexico, we better see pretty consistent results. And so it actually provides a great quality assurance procedure for us to be able to take advantage of using the same databases across multiple networks. But when we think about reporting out the results, it is quite likely that we will want to focus on reporting out only one instance of of results from a particular data asset. And so as Kristen's highlighted here, we're just going to make sure that we keep track of that as the results start pouring in. We've got a question from Chan, so you can tell us again where are the validated phenotypes. So the phenotypes that are in development right now are at atlas-COVID-19. However, the phenotypes that are for the final packages, we have not yet posted them to atlas.odyssey.org. We are still finishing our review for that. So that is really what's in queue for the next 8 hours, is to make sure that we finish those phenotypes, review them, and then push them into production. So I'm expecting that Christian and I are going to have a fun evening hanging out tonight as we go one by one through those cohorts and provide them to the community. Wojciech asked the question about where is this data sheet? So let's see if I can drag Teams over just to show you how to navigate to that. So in Teams, if you go to the competency study execution against network channel, this is the channel that Kristen and Talita have been leading. Under files, you'll see that there is a spreadsheet called Odyssey COVID-19 Studies Master List. And so you can enter this sheet and take a look at the data that we have. I'll also reinforce that Kristen and Talita and George and Jose have been doing a great job of trying to collect all of this data on behalf of the community, but I'm pretty sure that it's probably not even necessarily complete, because I believe I've had conversations with other members of the community who have access to data that are not yet on the list. So any of you who have access to data, if you're seeing yourself not listed there, please take a moment and put yourself in the sheet, or you can simply ask, contact Kristen, and she'd be happy to coordinate that on your behalf. We've got another question here. How is the community working to triage the study packages? We only have so much compute. Yeah, this is a great point, and this is really where we're going to be leaning heavily on Kristen and Talita in their work group to deal with this. So as each study package is prepared, we're going to make them available on GitHub and have them accessible for everybody to run. But Talita and Kristen have already started to outline the sequence of priorities that we want to execute. I think being respectful for the need and the support that we've been getting from our Korean colleagues, there's a clear need that we want to prioritize pushing study packages so that we can execute against the HERA data set. And we want to make sure that we prioritize running those characterization studies, because the results of the characterization studies will directly inform some of the decisions that would be subsequent to some of our analyses. And so we're going to try to kick those off first. But from there, we'd really like to be able to have the packages for patient level prediction and estimation available and ready to run. What's noteworthy about the patient level prediction packages is that there's going to be 2 stages to that. There's going to be a model training activity that will go on, and then there's going to be an external validation exercise. Now, model fitting, depending on your data size, can take quite a bit of time, but actually model evaluation, doing external validation, that's actually a quick job that can be run. And so for those data sources that are limited in terms of their compute, we'll probably suggest to you that you do not need to be able to do the internal validation. We could trust the models that are being built from the rest of the network, but we'll ask you to do external validation to make sure that our results are robust. For the estimation studies, we'll certainly be prioritizing the specific comparisons for which we have data. As we talked about early on, we've received high interest from the EMA, the FDA, the CDC, and from our colleagues in Korea about this question about ACE inhibitor use. And certainly based on the prevalence of hypertension and the prevalence of use of ACE inhibitors, that seems like a question that could have a high impact. But it's also very important that we look at hydroxychloroquine, which is currently seeming to get the most international attention as a potential prophylactic or treatment for COVID-19. And we want to make sure that we generate as much safety information about that product as we can. Subgroup analyses are likely going to be prioritized secondarily for those sites that are limited in their compute resource. Next question. For those of us with R but not Odyssey skills, can someone run a quick targeted tutorial so that we can help on that front, wherever the help is needed? Well, I didn't actually plan for this, but let's just do this. Whoa, whoa. Book of Odyssey is where I would go. In the Book of Odyssey, you'll actually be able to read about all of the Odyssey packages. And what was an amazing, amazing community effort to put this together was that you actually can see not just the theory and principles, but you can actually see the working code. In addition to that, all of our packages for prediction and estimation have really nice vignettes. Martijn did a really beautiful job of preparing for the cohort method and for the self-controlled case series method, step-by-step instructions that you can walk through to learn how to run those packages. And Jenna and Peter did the same thing with the patient-level prediction package. So I'd encourage you to read the Book of Odyssey and also take a look at the vignettes that are available inside of each of the GitHub repositories for all of the Odyssey packages. Wojciech had another question. So do we have a plan after the Study-A-Thon, e.g. recommended ways to ETL your data if you happen to have a significant caseload later? So this is a good question, Wojciech. This is something that actually George has been spending a lot of his time liaising with other data partners. We've heard a lot of enthusiasm from sites who have either done some starter work towards converting to the OMOP common data model, or have a desire to do it but don't even know how to get started. And I think one of the insights that we'd really like to bring from this Study-A-Thon experience is what ETL tips and tricks can we learn from the community that would help a new site, particularly as it relates to the nuances of the emergency data that's being collected for COVID-19, both in terms of diagnosis codes and testing procedures and laboratory measurements. But I think in general, as a community, I think this really can also provide a call to action for us to help each other with developing ETLs and providing tips and practices, but also just helping through the process. You know, with the Eden Academy being stood up and a collection of small and medium enterprises being certified now to be ETL supporters, I think we have a great international infrastructure to help new sites that come online to be able to get their data from whatever raw form it is into the OMOP common data model as quickly as possible so that they can start to participate in running these analysis. And so I certainly hope many of you will join the effort to help others in joining our community and participating in these studies. There's going to be a clear need for us to bring as much data together to answer these questions as we can. Next question is about the COVID-19 cohort evaluation code. So the question specifically, just to read it, is will the coming studies take advantage of the cohorts already generated, or will they be recomputed all over again? So the COVID-19 cohort evaluation package, this is a package that Martijn Schumi has developed. And what it does is it takes in a series of cohort definitions as input, and it allows you to perform the cohort diagnostic procedure on those cohorts to then report out the results. Martijn did something very clever with that package, which is that it takes as input a series of cohort definitions. If we add extra cohort definitions to it, it's smart enough to know which cohorts have already been run, and it will not repeat that. So if you've actually kicked off the code last night, you certainly have not wasted your time. All of those cohorts that you've run, most of those are actually the exposure cohorts, which have not changed over the course of today. And then the other big batch of them are the legend cohort definitions, which will not change. But what we will be doing is adding a series of cohort definitions into that package tonight. And if you kick off that job tomorrow, the package will basically notice the ones you've already finished and skip those, and only run the analysis for the cohorts that you haven't yet run. Just one more comment, and just to reinforce and to really go back to that ETL comment. It's very important for us to continue to reinforce. Yes, we're working super hard these couple days during this Study-A-Thon, but this is absolutely a marathon, not just a sprint. we're going to be working together for quite a while on this. And as it relates to data, we need to be thinking about not only what new data are out there that can be ETL to the common data model, but also how can we maintain the data that we're actually supporting within this network here. And so as Chan wants to reinforce the HERA data, he and his team are doing amazing work to update that data regularly. And so we're trying to keep it up to date as regularly as possible. I know the Columbia team is running their ETL every single night to make sure that we have the most recent data available for analyses. And so I fully expect we're going to be in a situation where we will be repeating analyses on a regular basis, not just in the next couple days, but for the many weeks to come until we feel like we've generated the evidence to support this issues. All right, I've got one more question, and then we'll call it a day here. Wojciech asked, if we have symptom onset data stated by a patient, do we track that? Historical temperature readings, cough now versus started 3 days ago. Do we mark the date as computed? So I think, Wojciech, you're asking questions specifically that are probably nuanced for every data source and how they're actually ETLing their data. I can tell you as it relates to what we've been doing in our cohort diagnostics, as we've looked across multiple databases, we've been looking very specifically at those symptoms to understand when are they occurring in what sequence. I can tell you myself, I spent a good portion of the day with Danny and Anna and Christian, trying to evaluate pneumonia and understanding when that takes place. And with this kind of idea that pneumonia is a complication that comes later, we kind of thought they would come later, but actually a good number of patients are just presenting initially with pneumonia. And so the question really becomes, what is the timing of these symptoms and how does that actually play out? So that's something that we're all going to need to learn about much more thoroughly as we go through the process of conducting these characterizations and performing our analyses. I see no more questions, so I want to Craig, can you tell me what you want me to say?
Just a reminder, because there's going to be some new people that, since you showed the Book of Odyssey, that they can get that. That's right on odyssey.org. If they don't have the beautiful book you're holding, they can get the link to both the English version and the Korean version on odyssey.org. So it's a great resource for help if you need it immediately.
So you got the English version, you got the Korean version. If you go to book.odyssey.org, you'll go straight into the textbook, but you can also get to both. It's a open source book that's posted on our GitHub, and you can also get to it just by going to odyssey.org, and you'll be able to link straight off of that. Thanks, Craig. All right, so I'll call it a wrap for our day 2 update number 3. I want to thank you all for your active participation and collaboration, and I look forward to seeing some of you in 8 hours. Thank you all very much.